Good morning. This video will be exploring about 4 different topics on just sort of interesting neuroscience related topics. Phineas Gage, Alzheimer's, HM, famous neuroscience patient, and split brain experiments. To start off with, we'll talk about Phineas Gage. If you wanted to Google it, Phineas Gage is spelled with a PH and Phineas. And once you have that, it'll be all the first page hits. So Phineas Gage had an accident back in the 1800s, in 1848 actually, and he worked on the railroad. And I'm not an expert on how railroad stuff works, but apparently whenever you are building a railroad, I did know that they'll like kind of blast their way through mountains and stuff like that. And so whenever they're trying to, blow something up, I guess you drill a hole, you put the explosive down in the bottom, and then in order to keep the explosive once you light it, like you put explosive and then a fuse coming out of the hole. And then if you were to just light the fuse, then the explosion would just come up out of the hole. It wouldn't go into the ground. So you put explosive, put a fuse, then you tamp down something on top of it like sand or dirt or whatever. And then when you light the fuse, now the explosion is more likely to go out and break up the rock that you want. So there's this guy named Phineas Gage, that's Phineas, and he had a tamping iron. So again, you put in there and you tamp down the sand with your tamping iron. His tamping iron is 1 1/4 inches in diameter. So something about like that. It weighed somewhere in the neighborhood of 13 pounds. And apparently he got distracted. Somebody said something to him. He filled the hole with the explosive, but then didn't put any inert substance on top of it, the dirt or whatever. And so when he hit it with his tamping iron, created a spark or something like that, and it exploded. And so his tamping iron went up underneath his cheek, out through his head, and then flew pretty far away. You can go on Wikipedia and however much you trust these accounts from over 150 years ago, supposedly the tamping iron was found somewhere around 75-80 feet away. On one hand, that sounds crazy. On the other hand, it's like I'm blasting the earth to help build a railroad. So I would think the explosions would be pretty significant. It's not, black cat firecrackers or something. So here he is afterwards. This is a confirmed photo of him. He lived. And so it, again, underneath his cheek, back behind his eye, so this is the pathway that the tamping iron took. Again, remember, 1 1/4 inches in diameter. And this is a photograph that was found of him with his tamping iron. Here's a few more renditions of the pathway where it went through the frontal lobe in his brain. And he lived for about 12 years afterwards and then eventually died. So in museums and such, I can't remember which museum this is at, but they actually have the original skull and the tamping iron next to it. You can find some books online about Phineas Gage. What's significant though is it went through his frontal lobe, and after that his personality changed. There's debate as to how much it changed. So almost like tabloids or news of today, if somebody writes it down a certain way, you can kind of slant something, right? With politics, if it was a Democrat or a Republican, then Fox News would take a different slant towards Republicans versus Democrats versus like CNBC or something like that. So my point is, there's debate as to how drastic it was, but it did change his personality somewhat. He lost his job and everything else. Supposedly, from moderate claims, it seems like his personality changed some early on, but then after a few years, he kind of got it back together and drove a stagecoach or something. But originally he was like a foreman, and he lost his job because he turned into kind of a jerk. So from here and from other experiments, we now know and believe that your frontal and prefrontal lobe, this part up here, is really what makes us human. From here back, we're largely just, giant mice. My cerebellum works the same way as a mouse's. I can see, I can hear, I can taste. just like a mouse. An eagle can see better than I can, a dog can smell and hear better than I can. So it's this front part. When we think of like humans as being intelligent, a lot of that is up here in our frontal lobe. And we think that our frontal lobe might kind of like keep our animal instincts in check. So for example, if somebody pulls in front of me in traffic, I might be like, man, I want to honk at that guy, or, I want to, give him a piece of my mind or whatever. But then my frontal lobe's like, you don't know this guy, is it worth it? I mean, you mouth off to some dude, we're in Texas, there's about a 20% chance that dude's got a gun in the car or something like that. It's not worth it. If you were to reach for your dog's food, at first he might want to bite to keep you away. But with a frontal lobe, your frontal lobe is like, don't bite the hand that feeds you. So humans have a more developed frontal lobe. So we're a little bit better able to control our animal instincts, in theory. This one, just for interest, Phineas Gage, Nicolas Cage. Okay, the next topic that we wanted to look at was Alzheimer's. So here's a slice. If we basically took a slice through this way of what your brain looks like, we can see the gray matter. In this picture, it looks kind of peach or pink in color. I educate America. And so you can see that part there, and then you can see the white matter. Well, you don't have to know these terms or anything, but there's like gyrus and sulcus. When you look at this, you see like hills and valleys. And so I'm just going to refer to them as hills and valleys instead of gyri and sulci. But you'll notice the hills and valleys right here, the valleys are getting bigger and the mountains or the hills are getting smaller. There's just less total brain mass. That's what happens with Alzheimer's. This is advanced Alzheimer's, as it shows. Here's an actual brain with Alzheimer's. So let me block something for just a second. Okay, so here's the back half of the brain. This is a person looking to the left over here, to your right if you're watching this video. And this is the back half of the brain. You'll notice the hills and the valleys. There's a whole lot of hill and the valleys are pretty narrow. So try to predict what the front half of the brain would look like. Now that it's revealed, you'll see that there's like hills and valleys. The valleys are getting a little bit bigger here, but there's still some big hills. Then all of a sudden here, the hills are just narrow. It's not supposed to be like that. The front part, the hills are supposed to be just as thick as the back part. So this is Alzheimer's. So Alzheimer's can degrade brain tissue all over the place, but it tends to kind of start in the frontal lobe and move back. Well, again, the frontal lobe is kind of where our intelligence rests. And so people with Alzheimer's, we tend to get Alzheimer's when we're older. If you have no other genetically significant abnormalities, about 50% of adults display Alzheimer's symptoms in their 80s. So half of us basically get Alzheimer's by the time we're 80, or in our 80s, I should say. And this is one of those things where it's like all of a sudden, you're 85 years old and your daughter or your son walks in and you don't know that person's name, and it's your son or your daughter, or you wouldn't recognize your husband or wife or something like that. So that's Alzheimer's. Interesting, as far as what causes Alzheimer's, we'll look at a few causes of Alzheimer's. Here's an Alzheimer's brain again. Interesting though, there's still a lot of debate as to what exactly causes it. A genetic link is related to chromosome 21 in humans. So trisomy 21 is Down syndrome. And the difference between me and a patient with Down syndrome is just trisomy 21. I have two chromosome 21s, one from mom and one from dad. But I am predicted to get Alzheimer's. I got a 50-50 shot of getting it in my 80s, like I said. If I have Down syndrome, I have a 50% chance of getting it in my 60s. So basically, if you have Down syndrome, you have an extra chromosome 21, on average, you get Alzheimer's 20 years earlier. So we think that there's something to that. There's something related to Alzheimer's on chromosome 21. Is it possible that there are other genes all over the place on other chromosomes that are also related? Sure. But we do know that with Down syndrome, you get Alzheimer's a couple of decades earlier. So this is severely, a severe Alzheimer's brain. Another one, you can see the hills and valleys in the back, and then look at how narrow those hills are in the front. Okay, so as far as what are the causes, or what do we notice? What might be the causes? So here's normal neurons. And in Alzheimer's, we notice two major differences under on a microscope slide. So they have these neurofibrillary tangles inside. It's basically cytoskeleton that's kind of growing out of control and sort of like sticking together, like coalescing together. Imagine if I had almost like, I don't know, one of those spools of yarn, and I buy it from the store, and it's got like 300 yards of yarn, and it's all like neatly spooled. And then I unspool it and I try to like put it back in there, and it's this giant like tangled mess. That's pretty much what these are. And typically it's related to cytoskeleton. There's this protein called tau, T-A-O. T-A-O? T-A-U? It's tau protein, T-A-O or T-A-U, I'd have to Google it. That's supposed to be kind of like keeping all the cytoskeleton in place, and it's not. So tau proteins, we think that messed up tau means messed up cytoskeleton, means neurofibrillary tangles, and now those nerve cells can die. We also notice these green things, amyloid plaques. Amyloid plaques is just made out of this protein called amyloid protein, and it just accumulates in the brain also. So what we don't know is Alzheimer's causing the amyloid plaques and the tangles, or are the tangles and the plaques causing Alzheimer's? We just know that when somebody shows Alzheimer's symptoms and they pass away and we dissect their brain and make microscope slides, we see these features. When somebody passes away without Alzheimer's symptoms, we don't really see these features very much, or they'd be much, much more mild. Here's what it looks like under a microscope though. This is a nerve cell. So you're looking down the barrel of a nerve cell. So let's say this is a long nerve cell, a neuron, and you're looking down the length of it like this. And those are the neurofibrillary tangles like shooting out. So it's actually spewing out cytoskeleton. It's a ruptured nerve cell, essentially, is what you're seeing. Now, part of the reason it's ruptured is because we like cut through. You're not supposed to have that much cytoskeleton leaking out. Here's a picture of how those tau proteins, the blue parts, are stabilizing cytoskeleton. Yeah, tau, T-A-U, tau protein. And then the last thing, these are the amyloid plaques. So under a microscope, we can stain them with this one specific stain, I don't remember what it is, and it sticks to those amyloid plaques. So that's an amyloid plaque, that's an amyloid plaque. And again, we have that linked with Alzheimer's. There's so much more stuff to do in neuroscience, but we'll look at, we'll look at this next. This guy you'll find interesting. For the longest time, I learned about him in AP Psychology my senior year in high school. I was a senior in the 93-94 school year. And so this is HM. He was anonymous for a long time, and then about a decade ago, I'd have to look up the date, about a decade ago he died, and so they released his full name, and it's like Henry Molaison. I think I've got it right here, Henry Molaison. Yeah, he died in 2008, so about 12 years ago. And HM was a peculiar case. When he was a kid, there are many books. There's one book I've got called Memory's Ghost written on him, but he basically got in a bicycle accident and then started having seizures. Well, if you have a seizure once a year, it sucks. When you're older, I don't know if you can get a driver's license, but otherwise, one day out of the year, it's kind of dangerous maybe, unless you can feel it coming on and sort of get in a safe place, but otherwise, 364, you'll probably be okay. But what if you have seizures every single day? What if you have a seizure, it's intense, and so afterwards you're exhausted, you nap for a few hours, and then the next thing that wakes you up is another seizure. So at some point, you might even do a risky surgery to stop the seizures, because it's not just an inconvenience, it's like wrecking your life. So they went in, and they were going to do a lobotomy. A lobotomy is where we cut or remove part of your brain. And so they did. The part that they removed is called the hippocampus. This is the actual brain structure, and that is a seahorse. Hippocampus means seahorse. So the hippocampus sort of looks like a seahorse, hence the name. As far as where is it in your brain, it's kind of tucked away in the middle a little bit. So this guy's looking this way. We kind of cut through his brain, and his hippocampus is this blue part right here that kind of loops around. And here's sort of the picture until you've seen, if I had like a brain model with me like I do at school, it'd be easier to notice this. But basically a normal brain, if I look up like that, this is the view of the brain that you're seeing. These are the olfactory bulbs that go into the top of your nose so that you can smell stuff. His, you can see where the cuts were made that removed his hippocampus and some other small parts of his brain. I'm going to tell you the significance of like, well, so what's so significant about HM in just a second. Actually, I'll tell you now. So he had this surgery and they removed his hippocampus. And so afterwards they wake him up, and there's some debate as to how much of it was accidental, like they accidentally removed a little bit more of his brain than they had intended. There was some sort of like cutting, but then a suction device involved. So I picture it kind of like when you're at the dentist and they're squirting in water, but they also have that little like vacuum suction thing that they're like sucking up the water in the spit. I'm picturing something like that. So anyway, he wakes up and, at first they're talking to him and he seems sort of out of it, but we expect that, your brain's going to be swollen and everything else. So after brain surgery, we expect you to not quite be yourself for a little while. And then they realized something later on. Day after day after day, he'd wake up and he didn't recognize the people that were new, like his current nurse. Maybe he'd been being taken care of by a nurse after surgery for the last week or something. He didn't recognize her, and she would visit him, hourly or something maybe. Well, it turns out they removed his ability to make long-term memories. So your hippocampus, we now believe, is the primary thing that converts short-term into long-term memories. So what that means is it's a little bit like a hard drive. So we got an old hard drive here as a visual aid, and pretend that all of a sudden this hard drive could no longer write or save any new information. My computer no longer had a save button. Everything that's already on here is there and I can access it. So let's say that I'm HM and I had surgery right now. I wake up. From this point forward, everything is a new day. It's Groundhog Day over and over and over again. But everything before I remember. So if you ask HM, where were you born? He can tell you where he was born. What was your mom's name? What was the address of your childhood house? Who were you friends with? Where'd you go to school? And all that. Well, that's already stored on his hard drive and he can access it. What's 5 times 5? 25. Ask him an algebra problem. He can do that. But I can no longer write anything new. So if you walk in and say, "Hi, my name's Bob," I can't store that anywhere. So when you leave and you come back 5 minutes later, I don't recognize you forever. So there was an MIT psychiatrist, his doctor after this, and her name is Dr. Conklin. I can't quite remember her first name, but it's Conklin, Conklin, if you wanted to Google it. And she studied him for decades, because this happened when he was fairly young, and he lived to be pretty old. And I want to say that she studied him for somewhere around like 4 decades, and he never recognized her. So she was his regular doctor, and she would have to reintroduce herself every single time, which is pretty significant. Can you imagine, like right now, the way that we think it works is short-term memory might last a few minutes. So if you just give me a random set of numbers, 5, 7, 8, 9 numbers, if I'm good, then I can remember those. And then if you distract me for like 10 minutes and I'm not rehearsing them or thinking about them or concentrating on it, if you distract me for like 10 minutes and I come back and try to spout them out, I have trouble because I didn't long-term memorize it. What did I wear yesterday? A black shirt. I know that because I saw it in the videos today that I was starting to upload for YouTube, that I scheduled to upload for tomorrow. What did I wear 9 days ago? I don't know. I didn't store that in long-term memory. 918-258-9838. That's my Broken Arrow, Oklahoma childhood phone number. We moved from that house when I was in about 8th grade. So I was maybe 13. So that was 30 years ago. So I haven't lived in that house for 30 years. And it was a home phone, so we didn't take it with us. but that is etched in my long-term memory. So my hippocampus said that phone number, 1801 South 4th Avenue, was my address. That phone number and that address, like that we store, but what did you wear a week and a half ago? I don't know. So I can remember something from 30 years ago, but I can't remember something from 10 days ago. Once he died, he was a nice guy. He basically was always really chill, and you can find a little bit of audio interview of him online if you're interested. But he said that he was happy to donate his brain to science. He was happy to be studied. Basically, if what I do can help you guys understand and help other people get over whatever medical problems they have, great. So he donated his brain to science, and they had an online event where they were shaving off his brain. They basically take the brain, and like I said before, the brain is not very firm unless you freeze it. Kind of like water. Water's not firm unless you freeze it. Then you can like slice through it. So they froze his brain. So there's his brain embedded in a block of, we'll call it ice. I'm not sure that it's like pure water. I'm sure it's some sort of pH balanced saline solution, whatever. And then they would just go through and just shave off slices of his brain. So they shave off a slice, straighten it up, preserve it, take a photograph, and kind of like, from what I can tell, sort of like laminate it, so to speak. And they just slowly shaved off his brain live on the internet. I actually watched a little bit of it. And this is just showing you what it looked like in the process of like shaving off little bitty bits of his brain. You can go online, and it's called something like the HM Project, I think, and they'll show you some of the significant images of the brain slices. So HM. And the last thing that I wanted to show you was what happens if you sever a corpus callosum or corpus callosum. I hear callosum probably a little bit more, so I'll go with that. So the corpus callosum, you might remember from a previous video, is my right brain and my left brain are on the tops, not really touching, but there's a set of this nerve bundle, sort of a nerve highway that connects the two halves called the corpus callosum. So my right brain, for example, controls my left hand, and my left brain controls my right hand. And so for them to talk about, to like coordinate a movement, like if you're going to throw a large ball at me and I have to catch it with both hands, then for me, my brain, I have an intact corpus callosum, and so I can coordinate right and left hand. But if you were to sever my corpus callosum, then my right and my left hand aren't working together as well. It'd be almost like, pretend that you're going to throw a football at me, and I've got one dude on this side, so here's me, Mr. Cotton. Then I have a student A right here and a student B right here. And we're like, okay, so student B, you're going to be the left hand, go, and student A, you're going to be the right hand, go. And then they have to coordinate to try to like catch a ball together. That's what it would be like if you had a severed corpus callosum. So there's a video that I'm going to put down as like an assignment, and it's about 10 minutes long, and this is what it starts out as. And it's basically a guy that has a severed corpus callosum, and they kind of show you some of the odd experimental outcomes of that. So what it comes down to, and I alluded to this a little bit in an earlier video, is this. Your brain is not symmetrical in function. In anatomy, it's fairly symmetrical. If you look at the right side and the left side, looks about the same, but from a functional standpoint, that's not necessarily true. So speech, as we mentioned before, Broca's area and Wernicke's area are in most of us on the left-hand side. Some people have speech on the right. There's some evidence that a little bit of speech might be on the right, but speech is overwhelmingly here on the left side of the brain. Body language, reading body language is on the right-hand side of the brain. So if this particular area that reads body language is damaged in you, you can't tell the difference. If I greet you and I'm like, hey, how you doing? Nice to meet you. Versus like, hey, how you doing? Nice to meet you. You can't tell the difference because you don't understand like body language, sarcasm, that type of thing. So what does this have to do with anything? Essentially, so I'm going to turn around this way so that my left side is also your left side. So here's my left brain. Left brain, right brain. So left brain is overwhelmingly the speaking part. And on my, well, we'll just say that. So my left is speech. And my left brain receives feedback and sends feedback to my right hand. So my left brain controls the right side of my body. It gets sensations from the right side of my body. So it goes back and forth this way. And my right brain controls the left side of my body, my left hand. It also senses left hand. So if I'm touching something with my left hand right there, my right brain is lighting up. If I decide to move my left hand, my right brain is lighting up. So you get into these weird situations where, and you'll see this in the video, they'll give this guy something that's familiar, like, I think I even used this example in an earlier video, give me an Expo pen. So if I'm blindfolded and you give me an Expo pen in my left hand, so here I am, you give it to me in my left hand. So which part of my brain is getting that information and feeling this? Well, for me, the information goes to my right brain. Because of the corpus callosum, I share that information with my left brain. So the information goes from my left hand to my right brain, and then my corpus callosum shares that information with my left brain. So if you ask me, if you put this in my left hand and ask me, what is it? I'll say, right. My right brain realizes that's a pen. It sends that information to my left brain, and I go, it's a pen. But what if you sever my corpus callosum? Why would you sever my corpus callosum? Well, if this is my brain, hang on, where'd the brain go? There we go. If this is my brain, and I have like epileptic seizures, then one thing you'll find is a lot of times seizures start in the same spot and then travel the same pathway. A seizure is just random electrical activity. So picture it like a thunderstorm of electricity in your brain and just like going everywhere. And if the part of my brain that controls my right arm is suddenly just randomly firing, then that's why I kind of do this weird like seizure convulsing type of thing. Not every type of seizure ends up with like full body convulsions. But if the part that controls my right arm is just randomly firing, then my arm is kind of randomly responding, randomly moving. So what we can do is a seizure starts here. Let's say it starts on the right side. It's there for like a second, and then it hops over the corpus callosum to the left side and spends like a minute there. Well, we could cut the corpus callosum, and now the seizure starts right here and then never makes it. So we're essentially sort of stopping the seizure as it's beginning. So that's what we do. So we sever the corpus callosum. So back to this. You put this pen, I'm blindfolded. You put this pen in my left hand, so here I am, you put it in my left hand like this, and the information goes up to my right brain, and you ask me, what are you holding in your hand? I can't tell you, because my left brain that speaks doesn't know. Okay, then, if you, ironically, gave me a pen, and I had to hold it in my right hand, and you said, what was it that you're holding? If you give me a pen and you put it in my left hand, and then say, what was it that you were holding earlier? I'll be like, so again, the reason that when you put the pen in my right hand to write, okay, so you give me this, and you say, what is it? And you give me a utensil to write my answer. Only my right brain knows the answer. My left brain controls my right hand, so it doesn't know the answer. So it's very strange. Corpus callosum experiments, severing the corpus callosum, is pretty uncommon nowadays. Now we just do it with pharmaceuticals, medicines, drugs. And so a lot of the patients that originally had severed corpus callosums that we have been studying for decades, a lot of them are dying. Not necessarily because of a corpus callosum, but just because they're old. So these experiments happened back in the day. But again, if you'll watch the video, it's on YouTube, and if you just Googled severed corpus callosum, you'll see it. It'll look like that. It is old. It'll basically walk you through, and like this guy right here has a severed corpus callosum. That's the guy, Gonzaniga, that's the doctor that does the experiments. He has a severed corpus callosum, and so they're talking to him. And here in a little bit, they set up some different experiments. Like for example, if you wanted to be a drummer, one of the things that good drummers are good at is I can keep one rhythm with this hand and a different rhythm with this hand. So for example, and I don't know jack about music. Let's say that this hand is playing in like a beat of 4, you know, like 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. And this one over here is in 4s, but it hits like every other one. So it'd be like 1, 2, 3, 4, 1, 2, 3, 4, 1, 2. You know, like 1, you know, like 1, 2, 3, 4, 1, 2, 3, 4. That's kind of easy. But if this one's in 4s, 1, 2, 3, 4, and this one's in 3s, 1, 2, 3. It's hard because I have an intact corpus callosum, so my left and my right brain are mixing them up. So let's say that this one was just supposed to go, if I was doing individual beats, 1, 2, 3, 4, 1, 2, 3, 4. And this one's supposed to go 1, 2, 3, 1, 2, 3, 1, 2, 3. And then maybe my bass drum, my foot is just supposed to go 1, da-da, 1, da-da, da-da, da-da. Each one of those is pretty easy. 1, 2, 3, 4, 1, 2, 3, 4. And this one's going 1, 2, 3, 1, 2, 3. And this one's going da-da, da, da-da, da. But try to do all 3 together, and your corpus callosum screws you up. Well, this guy, his corpus callosum doesn't screw him up as much. So if you assign him something to do with his right hand and something to do with his left hand, they don't mess up with each other. They'll do something in there, and this will be the last thing that we look at. I don't want to spoil the video, but they're basically going to do a challenge where they're like, all right, with one hand, draw like a shape like this, kind of a C. And with the other one, draw like a U. I'm right-handed, so I have a little bit more trouble drawing that. But I can draw a C, I can draw a U. But now try to draw them at the same time. That's me actually trying. And this one is messed up because my corpus callosum is sharing information back and forth between my right and left hand, and so that's why it's messing me up. It doesn't mess him up at all. So it's pretty interesting. So if you want to be a great drummer, just split your brain in half. All right, that's it. Hope you enjoyed the video, and thank you very much.